Okay, as I've said in your soft copy or in your module, I've already mentioned a very important person which is in the background, or which is in the discovery of nucleic acid, which is Mr. Friedrich Mescher. Okay, basically Friedrich isolated intact nuclei from cells and analyzing chemical content. Which they lead it with the extracted substances rich in phosphorus and nitrogen, and which basically he named it with nucleic acid. Aside from Friedrich Mescher, we also have Ivory MacLeod and McCarthy. They express it as a DNA and found as a genetic material by using a bacterial DNA to transform the genetic material of bacteria. So that is the discovery of Ivory, MacLeod, and McCarthy. So going deep with the discovery of Mr. Friedrich. Mr. Friedrich has actually identified the nuclein by isolating a molecule from a cell nucleus that will later become known as a DNA. So DNA behind the word DNA is actually the person which is named by Friedrich Mescher. So always remember that, guys, and please take note. Next is, let's go with the composition of the nucleic acid. As I've said, nucleic acid is a linear polymers of nucleotides, because nucleotides is the building blocks of nucleic acid that is actually basically consists of three components. We have the purine or pyrimidine, or it also called as a nucleobase, nitrogenous bases, the pentose sugar, and the phosphate group. As I've said, purine and pyrimidine is actually a part of a DNA and RNA. So basically the different of it is in purine, in the DNA part, we have adenine and guanine. The same in RNA, but in pyrimidines, in DNA, it is cytosine and thymine, and in RNA there are cytosine and uracil. Okay, and second to that, we have pentose sugar. Pentose is actually a monosaccharide, or a simple sugar with 5 carbon atoms. Always remember that. And the third one is the phosphate group, or it basically composed of phosphoric acid. So that is the three components of nucleotide, which is the building blocks of nucleic acid. Next, it also said that the nucleic acid is a polymer. A polynucleotide forms from the nucleotide monomers when the phosphate of one nucleotide bonds to the sugar of the next nucleotide. That is the reason why it's called polynucleotide, from one nucleotide bonded to another nucleotide and bonded to another nucleotide. So that is called as a polynucleotide, resulting to that a repeating sugar phosphate backbone with the protruding nitrogenous bases. So that is the composition of nucleic acid. Next, this is basically the summarization of the types of nucleic acid. We have the deoxyribonucleic acid, or the DNA. We have ribonucleic acid, or the RNA, and its division. It divided into 3 types. Okay, let's first go to deoxyribonucleic acid, or the DNA. It said that DNA is a double-stranded helical polymers of the deoxyribonucleotides found in the nucleus of the cell. Okay, what is the function of it? The storage of genetic material. So later part, we will be digging deep in the information about deoxyribonucleic acid. In contrast, we have ribonucleic acid. It said that it is a single strand. So that is the differences between deoxy and ribonucleic. In ribonucleic, it has a single strand, and deoxyribonucleic, it has a double strand. And always take note that the sugar of ribonucleic acid is called ribose, and the sugar of DNA is called deoxyribose. Okay? As I've said, it is a single-stranded non-helical polymers of ribonucleotides found in the nucleus and cytoplasm of the cell. So what is the function of ribonucleic acid or the RNA is to transfer a genetic information from nucleus to cytoplasm for protein synthesis. Later part, we will also digging deep with the information of ribonucleic acid. And of course, RNA is divided into three types. We have messenger RNA for the complementary copy of selected region of the DNA, and we have ribosomal RNA, which is along with the proteins forms the ribosome, which is the site of the protein synthesis. And lastly, we have transfer RNA or tRNA. It basically it's transferred the amino acid or the building blocks of the protein from the cytoplasm to the site of the protein synthesis. So that is actually the summarization of the types of nucleic acid. Going to the next slide. So now let's discuss what is DNA. Of course, DNA is very known in the medical field because DNA is a polymer of the oxyribonucleotides. So when we say polymer, it is basically defined as a substance or material consisting of a very large molecules or macromolecules. Why? Because DNA is one of the type of nucleic acid, and nucleic acid, as I've said, is also known as a polynucleotides, which is bonded to another nucleotides, or many nucleotides. That is why DNA is a polymer of the oxyribonucleotides. Okay, basically, DNA found in the chromosome, mitochondria, and chloroplast. In your soft copy, I've already gave you an unlocking of difficulties or giving you a background about the terms that you are not actually familiar. We have chromosomes. Chromosomes is the thread-like structure located inside nucleus of an animal or in the plant cell. And mitochondria and chloroplast, I know you've already know that in the prokaryotic cell and the eukaryotic cell. Basically, DNA carries the genetic information. That is why DNA is very important, because it carries genetic information or genetic material. Next slide. So let's have the components of nucleotides in a DNA. We have base, sugar, and phosphate. Always remember that every type of a nucleic acid, it has a components of nucleotides, and the component of nucleotides are those nitrogenous bases, sugar, and phosphate group. Okay, next slide. So this is actually the nomenclature of the nucleic acid. It summarizes the base, the nucleoside, the nucleotide, and the nucleic acid of the different nitrogen